Assalamu alaikum wa rahmatullahi wa barakatuh. Today's lab will be talking about flagellates. Previously, we reviewed the introduction into parasitology. We studied the classification of parasites. The classification included the protozoa and also helminths. Protozoa were classified into amoeba, ciliates, flagellates, and sporozoa. And previously, we also spoke of the amoebas and also we spoke of the ciliates. Today's lab will talk about the flagellates. There are many flagellate types of parasites. In here, we'll study two of them, the Giardia and also Trichomonas vaginalis. The intestinal, oral, and genital flagellates. Parasitic protozoa, which possesses a whip-like flagella as their organ of locomotion. The parasites are called flagellates because they have a long hair-like flagella, resembles hair, their body, and they use it for motility. The types of flagellates are divided into luminal dwelling flagellates, which are found in the lumen of the alimentary tract, and also hemoflagellates. They are found in blood and tissues. Most of the luminal flagellates are non-pathogenic. We will study Giardia and Trichomonas vaginalis, which are pathogenic. As for Giardia lamblia, it is endemic in the third world countries, developing countries. It comes alongside poor hygiene of contaminated food water. Water contaminated, not hygienic. Food also the same. It passes its life cycle in one host. The Giardia lamblia lives in the duodenum and the upper duodenum of the mucosa. It attaches to the duodenum and the upper duodenum of the mucosa by the means of sucking discs, ventral discs. Here are the ventral aspect where we see these sucking discs, where the Giardia lamblia, these are in the green color. It's called sucking disc. It attaches the Giardia to the mucosa wall of the duodenum and the upper duodenum and the small intestine. It is the only protozoan parasite found in the lumen of the small intestine. The morphology of it, the parasite exists in two forms, the troposoid and the cyst. We're not going to talk much about Giardia lamblia or any of the other parasites since it's a laboratory. We already spoke about it in lectures. Here we are just taking a summary. We'll talk about the images, what it looked like, how can we diagnose it. Simple. The main objective of the lab is you can differentiate between the shapes of the parasites. So, man acquires infection by ingestion of contaminated food and water. Direct person-to-person transmission may occur in children, homosexuals, and mentally ill people. Here is how the parasite goes through the human body with the drinked water. It goes through the small intestine. The mature cyst, the infective stage is the mature cyst. The mature cyst walks through the duodenum and then exsustation occurs. The exsustation is where the cyst turns into a tropozoid. The tropozoid has a rapid jerky movement, can adhere by ventral sucking disc to the mucosa of the small intestine. Then keeps its life cycle after divisions. It goes to encystation. Here, where the tropozoid turns into cyst. Also, then it finishes its pathway and gets ejected outside the body with feces. Cysts are found in feces. Tropozoid found only in the liquid stool. For the tropozoid is in the shape of a tennis racket. We've already seen it. Here it looks like a tennis racket or a heart. Here is the shape of a tropozoid in a slide. The slide is required of you. It comes in practical exams. Here is the shape of the tropozoid. Here is the shape of the cyst. The Giardia lumbea has two forms, the cyst and the tropozoid. For the tropozoid, it is the shape of a tennis racket, a heart shape, or pyrophone shape. Dorsally, it is convex, ventrally, it's concave with a sucking disc. The tropozoid is motile, resembling a fallen leaf. The motility of this tropozoid is resembling a fallen leaf, like a leaf falling from a tree. We have a link to a video where you can see the motility of the tropozoid under a microscope. Here we see the second disc. See this very fine line here around the middle, the upper half of the tropozoid is the second disc, where our two nucleus are in the middle of the tropozoid. They look like two eyes. They look pretty cute, actually. Here's the full tropozoid. Here's a ventral aspect and also a lateral aspect. The second disc goes through here, concave and convex. It is bilaterally symmetrical. Bilaterally symmetrical means it has two symmetrical halves. One half, this one half looks exactly like this other half, even with the number of flagella around the sides. It has one pair of nuclei, 4 pairs of flagella, 1 pair of exostyles, 2 sausage-shaped parabasal bodies. Here are the parabasal bodies. It has 1 pair of nuclei. Here are the red ones. They look like its eyes. 1 pair of nuclei, and the parabasal bodies look like mouth and nose, whatever you want to call it. It has 4 pairs of flagella. There's a pair, a second pair, a third pair, and a fourth pair. It also had an exostyle in the middle of it that divides it into 2. It looks like 2 pieces, it's like bifurcate. Okay, there is also a second disc. We also already showed you the second disc in these. This is the green area over here. That's a second disc. Here is the cyst. The cyst is different from the tropozoid. It is an oval shape or round. It has 2 pairs of nuclei. Parabasal bodies exist. Exostyle is over here. It has a thick cyst wall. It is a hyaline wall. It's a hyaline cyst wall. Hyaline is like clear, شفاف, clear, clean, شفاف. Hyaline cyst wall. The cyst, mature cyst, is the infective form of the parasite. The mature cyst is the infective form. الماتور cyst اللي هي عندها 4 nuclei هي الانفكتيف form اللي هي تدخل داخل الجسم وهي الفود, contaminated food and water. The cyst is small and oval and it's surrounded by hyaline cyst wall. قلناها تكون صغيرة وبيضاوية وعندها hyaline cyst wall. The internal structure includes 2 pairs of nuclei. A young cyst contains 1 pair of nuclei. By cyst is mature. There is a young cyst. It contains only 2 pairs of nuclei until it matures and the 2 nuclei becomes 4. The remnant of the flagella and the second disc may be seen in the young cyst. The remnant of the flagella and the sucking disc may be seen in the young cyst. Here, there are no remnants. Okay. The laboratory diagnosis is by stool examination. That is number 1. We can see and identify Giardia lumbea in the stool of patients with giardiasis. The identification of cyst is found. The cyst, we can find the cyst in the stool and the tropozoid in the diarrhea stool. In the formed stool, there are cysts. In the diarrheal stool, there are tropozoids. There is a test also called the Turo test or a string test, as they call it. Antigen detection, where we can detect the antigen of the parasite in the blood of the human being infected, and the antibody, we can detect the antibody to those parasites also in serum. Molecular method, where we can detect the DNA of the parasite by a method called polymerase chain reaction, PCR. It's a genetic method to detect PCR. Sorry, to detect the parasite. So the string test is a coiled thread, is a thread coiled inside a capsule. The capsule is weighted. A coiled thread inside a capsule, it is weighted. The thread is attached to the cheek and then swallowed. The capsule is then swallowed. The capsule passes through the stomach to the duodenum. After 2 hours, the thread is withdrawn, placed in saline, and is mechanically shaken. The use of interior test is not recommended for the high cost of the test. Here's the shape of that weighted capsule. We have two links here, the Giardia cyst under a microscope. The other link describes the Giardia tropozoid. You can see that online, clicking on it. The other parasite we'll study the other parasite. It is called the Trichomonas. The Trichomonas differs from other parasites. They only exist in the Troposoid stage. The cystic stage is not seen here. The genus Trichomonas has 3 species: Trichomonas vaginalis, Trichomonas hominis, and Trichomonas tenax. Here are images of the 3 species. They are very similar to each other, not identical, but are very similar. Here we will study only Trichomonas vaginalis. The Trichomonas vaginalis is encountered in all climates and all social groups, meaning we can find it any country and anywhere. It can live in all climates. So it is there. It affects people with multiple sexual partners or high sexual activity. In female, we can find it in vagina, while in males in the urethra. The Trichomonas vaginalis possesses 4 anterior flagella with short undulating membrane reaching up to the middle of the body. Here is an image of that Trichomonas vaginalis. Here is an image of a slide of it. It possesses flagella with an undulating membrane. These short strings, they are the flagella of it, while this long, rickety, zigzag line is called an undulating membrane. The exostylus bifurcates the Trichomonas vaginalis into 2 halves. It also has its nucleus. It's only in the tropozoid form. We have no cyst form. Flagella are characteristic. Flagella give characteristic webbing or rotatory motility. Webbing or a rotatory motility. It multiplies by binary fission in longitudinal axis. The image of Trichomonas vaginalis in practical. Here is this rickety line in here. It's the undulating membrane is very clear. There's a flagella coming from the top, nucleus is clear, and the exostyle is also there. The laboratory diagnosis of Trichomonas vaginalis, microscopic examination. What mount vaginal or urethral discharges examined microscopically? We have a wet saline where we place a drop on a slide and then we take a swab from the infected area. We swab it like wipe it with a cotton swab, and then we mix it with the wet drop and we view it under the microscope where we can notice that jerky or twitching motility and shape of the Trichomonas vaginalis. While the prominent stain, fixed smears may be stained with acridine orange, Nicola, and gemcisteins. We can stain it. We can either fix it and stain it, or view it under the microscope with a wet mount. The culture is recommended. The culture is considered the gold standard for the diagnosis or Trichomonas vaginalis. The accuracy of it is 95%. It is 95% sensitive. There is a serology testing for it and also molecular method. We said it includes PCR. Here is an image of that Trichomonas vaginalis. Another image. Here's a link where we can view the motility of it. The other flagellate we will study today are hemoflagellates. Hemoflagellates, they exist in the blood or tissue of humans, which is why they're called hemo. Hemo comes from the blood. Today we'll talk about Leishmania. All members of the genus Leishmania are obligate intracellular. Obligate intracellular meaning that they cannot live outside the cell. They live inside the cell, inside the like a microphage cell in the human body. So in the mammalian host, they multiply within macrophage, in which they occur exclusively in the amastigote form. So the Leishmania has two forms. The amastigote form, which is an image of it is here, so you can connect the amastigote form. You see it right in here. It's like oval or ovoid. It has an axon, a basal body, phenoplast, and nucleus. It's very clear. Here is the amastigote form. Here is the amastigote form in a cell of macrophage in the bone marrow. Here are the small amastigote form inside the cell. So the amastigote form is found inside the cell. It is intracellular. It found inside the cell of mammalian hosts. The insect vector female sandfly occurs in a promastigote form. This is the promastigote form of Leishmania donovani. It is spindle-like. It's longitudinal or spindle-like. This form of Leishmania donovani exists in the female sandfly. Here is the female sandfly. The Leishmania exists in the shape in the sandfly. This is the infective stage because we get the infection from the bite of the female of the sandfly. The Leishmania in the form of promastigote it goes through the blood vessel after being bitten by this insect. The insect works as a vector. The insect is a vector. It transmits the infection to the mammalians or human beings, to us, okay? The infective stage is the spindle-like promastigote form. The body is spindle-shaped with a single flagellum arising from the interior end. Here is the interior end, a single shape. Single flagellum. This is the nucleus, is very clear, and the phenoplast, basal body, and axon. Leishmania donovani. Previously, we were just talking about generally Leishmanias. Here, we'll specify to talk about Leishmania donovani. The images are required. Here's an image of Leishmania donovani, actually, in the macrophage of a bone marrow. So Leishmania donovani causes the visceral leishmaniasis or Kala-Azar. The disease is called Kala-Azar. The natural habitats of the Leishmania is reticuloendothelial system. They reside in the reticuloendothelial system, especially the cells of the spleen, bone marrow, and liver. So they cause the disease visceral leishmaniasis, Kala-Azar, or it's called dum-dum fever. This image is only to view. It is not to memorize. The symptoms are not to memorize. It's just explaining what the dum-dum fever is, the Kala Azar is, or the basal leishmaniasis is. Kala Azar, the same called as dum-dum fever. The symptoms include low degree of fever, hepatosplenomegaly, hepatos where the abdomen is swollen, severe progressive ataxia, swollen lymph glands, leukopenia, thrombocytopenia, monocytosis, loss of hair, and other symptoms. The disease is, if left untreated, it can be fatal. Here is a post-Kala Azar dermal leishmaniasis, those spots on the skin. So, Leishmania donovani exists in two forms: the amastigoid form, the flagellar form, where we spoke of, and also the promastigoid form. They exist in two forms: the promastigoid form or the amastigoid form. The amastigoid form is aflagellar form. It has no flagella, aflagellar. And the amastigoid form is the flagella. It has a flagella. This form, the spindle shape, exists in the human sandfly, while the amastigoid form exists in the human body or in mammals. So we get infected with this form, and then it enters the cell, the macrophage cell, and then turns into this. So the infective form is the promastigoid form present in the midgut of female sandfly. It gets transmitted to us by the bite of the female sandfly. Mode of transmission is the bite of an infected female sandfly, or vertically from mother to fetus, or rarely by blood transfusion or accidental inoculation in a laboratory. So the promastigoid form, which is found in sandfly and can be cultured, it's pear-shaped. The early state subsequently becomes long cylindrical spindle shape. Initially, it is pear-shaped, then it becomes long cylindrical spindle shaped. The nucleus is central. It has a chenoplast lies transversely near the anterior anterior end and consists of parabasal body and blepharoplasty. Blepharoplasty is the base of the flagellum. It has an axon, the vacuole here, the blepharoplast, parabasal body, and the nucleus. The flagellum projects from the front and may be the same length as the body of the parasite. There is no undulating membrane. No undulating membrane in the Leishmania. No undulating membrane in the Leishmania. Here is a slide of Leishmania culture. This form exists in a culture. Here they are dividing, splitting in two. Here is a single Leishmania. The laboratory diagnosis of the demonstration of Leishmania donovani bodies, amastigotes. Demonstration of Leishmania donovani bodies, amastigotes from the specimen of the blood, spleen, bone marrow, and liver. Culture may be done. Leishmania skin test is also called Montenegro test. Is based on the principle of light hypersensitivity. There are non-specific serological tests for it. The habitant, there's Leishmania tropica. We spoke of Leishmania tropica donovani. Next, we'll talk about Leishmania tropica. The morphology of Leishmania tropica is very resembling to Leishmania donovani. The habitat, amastigote and reticuloendothelial cells of the skin. The habitant is reticuloendothelial cells of the skin. Previously, we said that the Leishmania donovani's habitant is the reticuloendothelium, includes cells of the liver, the spleen, and also the bone marrow. The promastigote form is in the sandfly. The incubation of Leishmania tropica is 6 months. It causes disease called oriental sore. or Delhi boil, or Baghdad boil. Lesions are cutaneous meaning exposed. Here's an image of the cutaneous Leishmania, Leishmania tropica, oriental sore, Baghdad boil, Delhi boil. That is the size of it being measured. The laboratory diagnosis of Leishmania tropica is the smear from the specimen collected by puncturing the edge. Okay, so we see this lesion, or this one. We puncture the edge of it. We take a small sample and we view it under a microscope for diagnosis. Material may be also cultured in triple N medium to demonstrate the trauma-sigil form. The laboratory diagnosis is very simple. You could focus on puncturing the edge of the lesion to get the parasite, since it is intracellular. Focus on puncturing the edge of the lesion. That is it. That is it all about Leishmania tropica. It is very simple, and thank you very much for watching.